Hi, my name is Gurpreet. Our lab investigates mechanisms of lung inflammation and repair.
Hi, my name is Jessica and I investigate ozone-induced lung pathobiology by using fluorescent microscopic methods.
Hello everyone, my name is Manpreet and I study lung inflammation with animal imaging methods.
Lungs are continually faced with direct and indirect insults arising from sterile, such as ozone, and microbial components, such as the bacterial lipopolysaccharide or LPS. An overwhelming host response may result in compromised respiration and acute lung injury as characterized by lung neutrophil recruitment due to unregulated host immune, coagulative, and tissue remodeling. We describe a comprehensive analysis of various compartments, namely the broncho-alveolar lavage fluid, that is BAL, the lung vascular perfusate, that is LVP, left lung cryosections, peripheral blood, and the sternal and femur bone marrow. For ozone exposures, gently place mice in the custom induction box and continuously expose the mice to 0.05 ppm of ozone for 2 hours. For LPS exposures, anesthetize